Hello, my name is Dante Moore, and today we'll be talking about the determination of gender based on genes versus hormones. First, I want to show you a normal genome, specifically a male genome. We see 22 autosomes, or homologous chromosomes, and a set of XY sex chromosomes. The 22 pairs of autosomes, or homologous chromosomes again, are used to form our body and our characteristics. The sex chromosomes, on the other hand, direct the development of internal and external sex organs, or genitalia. In humans, gonads are the organs that produce eggs and sperm, which combine to form new individuals. In male, the organs are testes and produce sperm, and in females, there are ovaries, and they produce eggs. As shown here, sperm can be either X or Y, while eggs can only have the X chromosome. When an egg and an X sperm come together, a genetic female is made. And then when an egg and a Y sperm come together, a genetic male is made. It is not the combination of the XY, of just the XY though, that makes it a male, but rather specifically the Y chromosome that does. The X chromosome is important for allowing it to live, but does not affect male versus female differentiation. In some cases, there can be 3 sex chromosomes that are combined into XXX or XXY. Even in the cases of XXY, a male offspring is still produced. However, if it is just a Y chromosome and no X attached to it, then the embryo will be missing essential genes and therefore not develop into a human. Though when XX or XY is present, the embryo still has the ability to fully develop into a human with fully formed internal and external genitalia. The reason that the Y chromosome leads to the male sex is because of the sex-determining region of the Y chromosome, which is also known as SRY, the SRY gene. When the SRY gene is present, it causes the production of testes-determining factor, or TDF, that binds to DNA and activates additional genes which lead to further development of male genitalia, such as the testes. This is again shown over here as we see the Y chromosome with the SRY gene into TDF and then eventually testes. Now, differentiation of male versus female does not begin until week 7 of development. And before this, the embryos cannot be morphologically differentiated as male or female. As shown at the top, before week 7, gonads, as well as both Wolffian and Mullerian ducts, are found. Gonads are made out of an outer cortex. So the gonads are made out of an outer cortex and an inner medulla. And depending on developmental signal, the gonads can help to form specific genital organs. As we talked about in the last slide, TDF is formed in the presence of the Y chromosome, with the SRY gene. This factor is able to bind to DNA, as mentioned above, and activate genes that lead to the development of the medulla of the gonads into the testes. Once the testes are differentiated, they can start secreting hormones that influence the development of internal and external genitalia. The 3 main hormones secreted are anti-Mullerian hormone, testosterone, and dihydrotestosterone. In regards to the ducts, the development continues. As development continues, one regresses and the other one develops to form genital organs. As shown here in males, due to the production of anti-Mullerian hormone, the Mullerian duct will end up regressing. And then, due to the testosterone, the Wolffian duct is able to evolve to form the epididymis, the vas deferens, and the seminiferous tubules. Seminal vesicle. Here, we see the male external genitalia and its development, which is primarily due to the presence of DHT. As we can see, the genital tubercle when DHT is present, it causes the genital tubercle, urethral groove, urethral fold, and labioscrotal swelling to all form their specific male external genitalia. The genital tubercle leads to the formation of the glans penis. The urethral folds and grooves form the shaft of the penis, and the labioscrotal swellings help to form the shaft of the penis and the scrotum. On this slide, we see both internal and external female development. Originally, female genital development was thought to be very simple, based almost entirely on just the absence of the SRY gene. Although female embryos do lack the SRY gene, there are multiple genes in the X chromosome that are able to be involved in the development of the genitals. To begin with, yes, mostly the lack of the SRY gene is what causes the cortex of the gonads to become ovarian tissue, though there are some underlying genes that aid this process. Additionally, because of the lack of the SRY gene, there is no testosterone produced. The lack of testosterone causes the Wolferian duct to generate, and then also, because of the lack of the SRY gene, there is no anti-Mullerian hormone produced. So in the absence of this anti-Mullerian hormone, the Mullerian ducts are able to develop. These become the upper Mullerian duct, or the Mullerian ducts are able to become the upper portion of the vagina and the uterus and the fallopian tubes. Then, opposite the male, because of the lack of the SRY gene, no DHT produced, and without DHT, the external genital organs take on female characteristics, as seen over here. The genital tubercle forms the clitoris, the urethral folds and grooves form the labia minora and the opening of the vagina and the urethra, and the labioscrotal swellings form the labia majora. As I have shown, the determination of gender is based on both genes and the hormones produced through the development of the human. Specific chromosomes can lead to different hormone production, but also varying hormone levels among males or females will cause differing effects on the appearance and results of gender development. This continues after birth, throughout puberty, and then beyond that, and can have many effects on the different aspects of each human's life. So it's both genetics and hormones are incredibly important in the development of male and female genitalia throughout their entire life.